Okay, okay. Now we will take colleagues or participants to meeting hall. Now waiting in the meeting hall. You can see all of them. Now there are 28, 30. Maybe it's increased. There's a number of attendants. Some of them waiting room. Okay. Seher, başlayabilir miyiz?
Başlayabilirsiniz hocam. Kolay.
Okay. Okay. Buraya sen mi başlatacaksın?
Ben kaydı başlattım hocam.
Continue diyor ya, ben basayım bak. Evet, biz başlatalım. Dear colleagues, welcome to Professor Joachim Fandrey's conference. The topics of today's presentation: how cells sense and adapt to oxygen sensitivity. Before starting the meeting, I would like to inform you the academic life of Professor Joachim Fandrey. He got a doctor med degree from University of Lübeck in 1999, and then worked as postdoc fellow at University of Lübeck and Harvard Medical School. After that, he studied 4 years at Bonn University. Then he got assistant professor for physiology at University of Lübeck, and he has been working at Duisburg-Essen University Physiology Institute since 1999. Now he is head of Physiology Institute and acting director of Physiology Chemistry and vice dean of Faculty of Medicine. He has many national and international awards, and his 214 academic studies have been published high-level journals. There are 8,556 citations to this publication, and the ASH index of publications is 52, according to Scopus. Professor Fandrey and his research group focusing on issues related to oxygen sensitivity, such as activation of a transcriptional factor, Hypoxia Inducible Factor-1, hypoxia and inflammation, cancer, role of HIF-2 in brain injury and regeneration, and the protein-protein interaction of hypoxia inducible factor, and also another topics for Professor Fandrey, fluorescence resonance energy transfer technology. Now, I want to give just a little bit information about our presentation topics. Oxygen sensing allows cells to adapt their metabolism to low oxygen levels. It has a central role in the mechanism of many diseases, such as anemia, cancer, and some neurological disorders. It is essential during fetal development and placenta metabolism. And studies has been focusing on discovering new drugs that can interfere with different disease states by either activation or blocking the oxygen sensitive mechanism. And when we look at this area, we can see in this field, 3 subjects were crowned with Nobel Prize. First one is the 1938 Nobel Prize in Physiology or Medicine to Cornel Hermans, awarded discovery showing how blood oxygen sensing. Then second one is Nobel Prize in Chemistry 2004, shared by 3 academicians who made fundamental discoveries concerning how cells regulate the breakdown of cellular proteins Also, this mechanism is great importance for regulation of hypoxia inducible factor-1 alpha, and Professor Fandrey will give detailed information that topics. And this last study opened a new way to understanding the oxygen sensing, metabolism, or processing. Last one Nobel Prize related to this area. The Nobel Prize Physiology or Medicine was awarded jointly Killen, Rockfield, and Semenza for their discoveries of how cells sense and adapt to oxygen availability. They established the basis of our understanding at how oxygen levels affect cellular metabolism and physiological function. Professor Fandrey will provide us very valuable information on the subjects, which includes the most basic molecular mechanism in the field of health science, including his own experiences. I think that Professor Fandrey's presentation will give us stimulating and inspiring information to us. Now, after this information, I want to give microphone to Professor Joachim Fandrey. After presentation of Professor Fandrey, we will take 3 questions directly, and after that, if we have more question, you can write directly to Professor Fandrey. It is possible. Yes, Joachim, microphone is yours.
Yes, thank you. Thank you, Orhan. Thank you very much for the kind introduction, and thank you very much for the invitation. Dear colleagues, I'm very happy that you found the time together and listen to my presentation. And I think I will start now to show you my screen. Just setting this up. Okay, so okay, so I hope you can see the full screen now.
Yes.
Yes, okay.
Yes, Joachim.
Okay, perfect. If I become too fast, please stop me. I have not too many slides, and I've scheduled this for 45 minutes so that we have time for discussion. But if it gets too fast, then please stop me, and then I just go slower, and then then we have questions in the end, and I'm happy to answer any questions and go back to the slides and so. So you have seen the title of my talk, which is actually the second half of this quotation, which is from the Nobel Committee in 2019, where the Nobel Prize for Medicine or Physiology was awarded. And the fundamental finding for which these 3 scientists, I will show you in a minute, received this award was how cells sense and adapt to oxygen sensitivity. And that is the title of my work. The title of my whole scientific work, more or less, I would say 95%, and also of this presentation. And I was very fortunate to meet these 3 gentlemen and to know them personally. And I may even say at least 2 of them are good friends of mine. And they are here in alphabetical order. Bill Kalin Jr. From Boston, from the Dana-Farber Institute at Harvard University, Sir Peter Ratcliffe from Oxford University in Oxford, and Greg Semenza from Johns Hopkins University in Baltimore. All 3 are fantastic characters, and before this terrible pandemic came over us, we had a Keystone meeting on hypoxia in Keystone in the beginning of 2020. which was right after the Nobel Prize was awarded. And it was such a fantastic meeting because all three were present, and they all three gave their view on oxygen sensing, on what they did. And this was a fantastic meeting. We also had Peter Ratcliffe at the Europhysiology, so the European meeting of physiology in London, 2018, where I was the chair for the German Physiological Society. And if you want to hear one of the very nice presentations from Peter Ratcliffe, this is in the web, is available. So you find this on the Europhysiology as a YouTube video. That's also a fantastic presentation. So I will want to try to guide you through the story of oxygen sensing by the work these three persons did. And for that, I will use an overview scheme, which I made many years ago, which essentially shows the process of oxygen sensing. And I will come back to this slide and this figure and then go through the different steps as we know today what makes oxygen sensing in the cell. So just for explanation, we have the two situations, a high oxygen concentration and a low oxygen concentration called hypoxia. And again, the two, to define them by partial pressure of oxygen is an interesting question in a lecture by itself. We may come back to that in the end. We are here speaking about the genetic response to hypoxia, which in the end causes expression of genes, and they are controlled by an hypoxia response element that is a DNA element. And to this element, proteins, the HIF, the Hypoxia Inducible Factor transcription factor complex binds. And so what we now have to explain, and that is what the three scientists did, and where we also try to contribute, is how is this transcriptional activation organized and initiated by changes in a physical number in terms of partial pressure of oxygen. And that is the fundamental question that we addressed and I addressed during my scientific career. Now, the different contributions by these three Nobel laureates can very nicely be focused on these three parts in the slide. So Greg's are contributing to the regulatory elements, the hypoxia responsive elements that control gene expression by low oxygen. Peter Ratcliffe contributing fundamentally to the process of the oxygen sensor itself. So the enzymes that use oxygen and transduce this into altered modified proteins that are then signals in the cell. And Bill Kalin, who fundamentally contributed to the process, how these proteins are regulated so that they are available for inducing the transcriptional response. Now, my start was as a postdoc in Frank Bunn, so Professor Frank Bunn at Harvard University in hematology, where I spent my postdoc at the beginning of the 90s. Frank Bunn is known as a hemoglobin person. And he, for example, he described the glycosylation, non-enzymatic glycosylation of hemoglobin. So hemoglobin 1AC, we all know for diabetes control. And in his lab, I started to set up a method where in animals, so these are rats, we could quantify the mRNA levels of erythropoietin mRNA. So erythropoietin is the hormone that controls red blood cell production. And if the body becomes hypoxic, such as at a low hematocrit, so hematocrit normal values would be around 40, also for the rat. And if this hematocrit, so the percentage of red blood cells in the blood gets low, then the tissue becomes hypoxic, low oxygen, because there is no oxygen transported to the tissue. And this is kidney tissue. These are the organs where the erythropoietin is made. And then there's an exponential increase in erythropoietin expression. And this is then followed, this is serum erythropoietin of the protein. The protein is then secreted to the bloodstream and is traveling to the bone marrow where it stimulates red blood cell production. So regulation of EPO production was the fundamental question of my supervisor, Professor Jeltmann in Lübeck, where Orhan also spent some time as a postdoc and where we met, as we said, 24, 25 years ago. So erythropoietin is exponentially increasing when you lower the hematocrit, and this is preceded by direct transcriptional activation of the gene. Now, this is unusual because if you have not enough oxygen, usually 2/3 of the proteins in the cell are reduced in production. So that the expression of a gene is increased was in those days somehow new and interesting, and it was understood as an emergency response to oxygen deprivation. Now, to study this in the whole animal is rather difficult, and we therefore used a cell model which was then developed in Frank Bunn's lab, which is a hepatoma cells. Erythropoietin is also produced in hepatoma cells. And these cells then could recapitulate the situation. And now we have here precise oxygen tension in torr, so millimeter of mercury. And when we decrease the oxygen tension in the cell culture, this was measured and then well controlled, we also have the exponential increase in the transcription of this gene. So clearly indicating this is a cell autonomous process. So one cell can sense the oxygen tension and can translate this in an increased gene expression. The big question is, how is this be performed? How can this happen? A physical number, oxygen tension, and a genetic response mRNA expression. And here comes Greg Semenza's early work. So Greg Semenza was in those days, we are now speaking of the early 90s. This is one of his seminal fundamental papers. He was in the Johns Hopkins in the Department of Genetics. Greg Semenza is a pediatrician and geneticist in the lab of Dr. Antonarakis. And he made this effort. He used the erythropoietin gene, and he was scanning the regulatory elements 5' upstream of the EPO gene and downstream of the EPO gene. And he made transgenic mice, many transgenic mice, where he introduced the erythropoietin gene and regulatory elements. And by this tedious work, he found that there is erythropoietin expression induced by a liver inducible element. There is a negative regulatory element, and there is a kidney inducible element, which is far upstream here and probably also down here. That was his assumption. Nowadays, we know that is several kilobases upstream. But the fundamental finding was that he found regulatory elements that control the erythropoietin gene under hypoxia. And that was proof that it's really genetic regulation. Fundamental finding, early 90s, molecular and cellular biology, in those days, one of the top journals in molecular biology. So, and he went further, because if you have these DNA elements, you need something that turns them on. So he used a method, and I don't know whether any students, PhD students are present. They probably have already forgotten this method. So this is a method which is called electromobility shift assay, where you use a radioactive labeled DNA probe, where you put in a radioactive phosphor, and then you mix this piece of DNA, and this piece of DNA was from the elements, from the sequence Greg Semenza had identified that they are enhancer elements for the EPO gene. So he used these DNA elements, and then he mixed them with nuclear extracts, with nuclear protein extracts. And what you find here are shifted bands. So the shift is from here to here. The mobility is reduced because proteins bind to the DNA. And this is normoxia, this is hypoxia for the liver, normoxia, hypoxia for the kidney. So he clearly should show that something is induced under hypoxia, and this is a nuclear factor, and therefore he coined this protein hypoxia inducible factor. This is actually something when you sometimes read or review papers today, and people speak about hypoxia-induced factors. No, that's wrong. The original nomination by Greg Semenza was this is hypoxia-inducible. So this is something that is, as he thought, already in those days something that is present, and by hypoxia it is induced to bind in the nucleus to the regulatory DNA element. And that was fundamental because it gave it made possible to isolate this protein. Again, this is easier said than done, because what Greg Semenza, or better his very, how should I say, hardworking postdoc Guang Wang in his lab, he used a piece of DNA and went on fishing. So he fished through liters of nuclear extracts. And if you have ever made nuclear extracts, you know what size of culture dish you need to make some milligram of nuclear protein. And then he fished from this nuclear protein by binding to the DNA. He fished the proteins, and they were sufficient so that he could then come with a sequence. And he found that hypoxia inducible factor has a classical structure, which is basic helix loop helix, PAS. I come to you in a minute. And that this protein was already a heterodimer and regulated by cellular oxygen tension. So oxygen tension regulation is important because it's hypoxia inducible. This structure links the protein to a family which has basic helix loop helix, is something to bind to DNA. That is a basic part of the protein that interacts with the major groove of DNA, so of desoxyribonucleic acid. And PAS was something, a domain that was known from other proteins, from period, which is a clock gene in Drosophila, from the aryl hydrocarbon receptor, which is the dioxin receptor, it's another name for it, and for single-minded, another Drosophila gene, which determines the orientation, the sides of Drosophila flies. So this was part of a family. And then the very interesting thing that came out was that it is a heterodimer of 120 kD HIF-1 alpha subunit. And as we will learn in a minute, this is the oxygen sensitive, and a constitutive smaller subunit HIF-1 beta. And this one was already known. This was called ARNT, for aryl hydrocarbon receptor, that's the dioxin receptor, nuclear translocator. So in those days, people thought this protein is responsible for the dioxin receptor to enter the nucleus and initiate the xenobiotic response in liver and so on. So again, from this initial finding, that's a heterodimer of a constitutive factor and an alpha subunit that is regulated by oxygen sensing. These with the other two papers are the seminal papers that made him a Nobel laureate, because he identified this factor. And as you see, this is now about a year or so old. This was around the Nobel Prize. So I'm sure this will have many more citations now. So this is a classical, a citation classic. Now, this was Greg Semenza. And then the race went, or the race started, because the Radcliffe lab was already working on erythropoietin regulation. Peter Radcliffe is a trained nephrologist, and they had a fantastic group with two other postdocs. One of them is Christopher Pugh, which I interestingly met as a senior house officer when I was in London and was doing my final year in neurology. So he was then switching to nephrology, and Peter Radcliffe and Chris Pugh, they were interested in regulation of kidney function, and erythropoietin is produced in the kidney. And so they were very interested in how erythropoietin is regulated. So this is why they were very fascinated by the initial work from Greg Semenza and continued to work on this. And when they found, or when they used this DNA element, the hypoxia inducible factor binding domain, the HRE, they showed that this is the alpha subunit is the regulated subunit, and the beta unit is constitutive, so that only one half of the dimer is responsible for the whole complex to accumulate under hypoxia. And I can now come back for another time to my cartoon. So I have shown you so far the Semenza work here. And we are now here at this part where we see that the HIF-1 beta is always in the nucleus and is not regulated by oxygen, but the alpha subunit is regulated by oxygen, and the alpha subunit is even entering the nucleus through the typical entrance mechanism. This has all now been resolved. So the HIF-1 alpha protein is in the cytoplasm and is then under hypoxic conditioning entering the nucleus, forms the dimer, and the dimer binds to DNA and then drives transcription. And I will come back to our own data for more detail. Now, how is the HIF-1 alpha accumulating under hypoxia? And that was the work where the Radcliffe group found that VHL is involved. Von Hippel-Lindau protein is a known tumor suppressor protein and is known in several kidney diseases where cysts are formed. So this is why the nephrologists were interested in this protein, and they had observed that sometimes when mutations in the protein were present, the erythropoietin levels were very high in patients. This was observation in persons. So this is the third person in this lab, Patrick Maxwell, Chris Pugh, Peter Radcliffe. In my opinion, these are the three ingenious guys in Oxford. So they found out that VHL is involved in regulation of hypoxia inducible factor. And how did they do this? Now, they had renal cell carcinoma cell lines. And as I said, cysts and renal carcinomas are formed if the VHL tumor suppressor gene is mutated. So if you have a cell line with a mutation, this is a patient cell line, and you use the two conditions, normoxia, hypoxia, typical hypoxia-inducible genes like VEGF or glucose transporter 1 are not regulated. They are always expressed. And then they made the transfection of VHL. So they repaired the cell. They brought VHL back into the cell. And then they saw that the normoxic signal, the normoxic expression of this HIF gene is down, and there is an increase. This is moderate, but you will see stronger results in a minute, or we can go for this lactate dehydrogenase, where you see now in the repaired, reconstituted carcinoma cells plus VHL, there is normal regulation by normoxic low, hypoxia high expression. And if the cells lack VHL, then there is unregulated maximal expression of the HIF target gene. So this was clear indication that cells that have no VHL, something is missing. And as you have already seen in the cartoon, the missing link is VHL because you need this for regulation, low under normoxia, high under hypoxia. And the Oxford group went one, the Oxford group then came into, them got some support. They actually always understood this as support, because von Hippel-Lindau protein was a protein that was heavily investigated by Bill Kaelin in Boston. So Bill Kaelin is a tumor biologist and an oncologist, and he had searched all his life for different diseases with VHL mutations. So immediately when Bill Kaelin became aware of this work, he started to look for von Hippel-Lindau protein mutations and cells with mutations, and he found that von Hippel-Lindau protein is a negative regulating hypoxia-inducible factor. So exactly what I said in the last slide, if von Hippel-Lindau protein is missing, hypoxia-inducible factor is fully activated without regulation, and you need von Hippel-Lindau protein for proper regulation. And that now became a race. Who's the first to understand how this regulation is working? And this also then came out in very prestigious journals. And in fact, as you can see, this was from the very beginning until the Nobel Prize was a very fair race, because they found the same mechanism and published this back-to-back in Science. This was from Mircea Ivan, postdoc in Bill Kalin's lab, the first 4 pages, and the following 4 pages from Peter Radcliffe's group. Panu Jakula is a Finnish postdoc who's now back in Finland. And they did this work independently, which is always very important, because then you can rely that it's really reproducible and that it's true. And they both came from the von Hippel-Lindau role, that von Hippel-Lindau is important. And von Hippel-Lindau is a ubiquitin ligase I show you in a minute. And the targeting of HIF was through binding of this protein to HIF. And the question is, when does it, or why does it bind, and what are the mechanism to mark, to tag the HIF for binding? And they found that this is a hydroxylation of a proline residue, so prolyl hydroxylation by oxygen-regulated enzymes. This is now cutting a very long and very heated and very competitive story short, because initially many people thought that some phosphorylation would be important, including the groups in Sweden at the Karolinska, who always favored the tyrosine phosphorylation. And in the end, it turned out that it's a proline that is hydroxylated, so a different mechanism. Now, if we come back to our scheme, we now have already the main regulatory elements. So we have the HIF protein, and we have here hydroxylation within a certain domain. I will come back. I will explain these abbreviations in a minute. So here are proline amino acids, and these are hydroxylated by enzymes that are called prolyl hydroxylases. And these prolyl hydroxylases require oxygen. And so only under normoxia, when sufficient oxygen is present, then the hydroxylation can take place. The HIF is hydroxylated, and the hydroxylated HIF is recognized by von Hippel-Lindau protein, and this is then ubiquitinated, polyubiquitin chains, and is then degraded in the proteasome. So this explains why, if the VHL is lacking or is mutated, then the HIF protein is not degraded, and you have the full unregulated response. And it also explains how oxygen is brought to this process, because these enzymes require oxygen for activity and for hydroxylation. And they actually went one step further by isolating these, and this is one of the major contributions of the Radcliffe group, in addition to the VHL, is that they isolated these enzymes, because they in Oxford had a very productive group that was working with C. elegans, so with worms. And they were looking for worms. They screened these worms for a mutation, and they found the Eagle9 mutation. And this Eagle9 mutation is a dioxygenase. It uses oxygen, and it can hydroxylate proline residues. And so this slide is also an example how sometimes the race is heated, and how sometimes only a few months or weeks may determine whether you're part of the Nobel Prize or not. Because similar work from a different aspect by genetic screening was done in Steve McKnight's lab in Texas by Rick Brook, in those days a postdoc. And they were just by a month or so too slow, because this paper was out in Cell in October 5th. And in fact, when the paper was accepted, the Oxford group was having a party, and using the quotation from the Apollo 9 mission was, "The eagle has landed." So Eagle 9, this is the famous saying from this party when this paper was accepted, because they knew they have the oxygen sensor. They have a structure that can use oxygen to translate this in a protein modification, and that has then impact on the oxygen sensing process in the cell. And there was one thing which was recently, or which is now very important, because when they isolated this enzyme, this is something that made this more than just a description of a biological phenomenon. They understood the system. So they saw that these enzymes require 2-oxyglutarate. This is a co-substrate with hypoxia and with a HIF protein. And if you replace this co-substrate 2-oxyglutarate with a simply modified dimethyloxylglycine. So this is a glycine with an oxyl group and 2 methyl groups that is tissue permeable, can go into cell permeable. This is a pseudo substrate. So this one is used by the enzyme once and then blocks the enzyme. So this is an inhibitor of HIF hydroxylases. And this is the initial description of a prolyl hydroxylase inhibitor, which I will show you in a minute, because this is now used in therapeutics already. So to modify the activity of a system. Okay, so now essentially this is the oxygen, this is the oxygen sensing process as we see. And the prolyl hydroxylases, there are 3 isoenzymes. I will show you in a minute a bit more. And so this is then essentially the basis, because these inhibitors can now used for drugs to stimulate erythropoiesis, because they stabilize HIF, and the HIF protein can then drive erythropoietin production. I come this back in the end of my talk, and this is the initial announcement of the Nobel Prize, as I was quoting that. Okay, so now a bit to what we contributed to the field, and this is in part also unpublished work, because these are the many processes that are regulated by hypoxia, and what you see here is a change in PO2 affects the metabolism of the cell, vasodilation, angiogenesis. We know this from physiology, but between gene expression and hypoxia, that was the question of the miracle. You now have learned that this was resolved by the 3 Nobel laureates. And in central is hypoxia inducible factor. So we made efforts to understand this better on the molecular level. Now, so far, I only showed you HIF-1 alpha. There is a second isoform, HIF-2 alpha, and also a third form, which I will not talk about, which is less studied. But HIF-1 alpha and 2 alpha are very similar in regulation, and they are oxygen labile, as I showed you. And the partner for both of them is the always in the nucleus available HIF-1 beta. So there are parts, oxygen-dependent degradation domain. This is the part that is important for the oxygen-dependent regulation. And there are transactivating domains where additional factors bind to gene expression. And here's the DNA binding. So the first thing what we studied is this part here of the degradation process of the HIF protein. And I only want to show you some of our figures, of our original data, which show you that this is still a very mysterious process. Let me briefly show what this is. These are hepatoma cells. This is under normoxia. This is immune histochemistry for HIF. In these pictures here, you see the colored structures is the nucleus with a HIF-1 alpha protein stained by immunohistochemistry. So normoxia, and then you put the cells 4 hours in hypoxia, and then you have nuclear accumulation of the HIF-1 alpha factor. And then you switch back to oxygen. And now comes the interesting part in the regulation, because what I've shown you so far is that the HIF protein is degraded through von Hippel-Lindau protein. And that is shown here because 3 minutes, so 1 minute, 3 minutes goes to 5 minutes, 7 minutes. You see when the oxygen comes back that the protein disappears. And if you calculate the half-life is around 5 minutes. So the protein, the HIF-1 alpha protein, is degraded very, very rapidly. It's a very unstable labile protein. And then after 14 minutes, the cells, or here after 30 minutes, the cells are cleared. You may even say the cells are, there is less protein in the nucleus than in the cytoplasm. That's something we are currently studying. So the HIF protein is degraded very rapidly. So the whole regulation I've shown you so far, hypoxia inducible factor, means the protein is, if a lot of oxygen is available, the enzymes are active, the von Hippel-Lindau protein is binding, and the protein is degraded. And that is what I showed you in the reoxygenation experiments. So if oxygen becomes available, the protein is degraded. If there's no oxygen, hypoxia, the protein will stay stable, it is not modified, and it can enter the nucleus and form the transcription complex. So that is a very smart mechanism, because the cells are now in danger. They have no oxygen and they do not want to produce new protein. Instead, they only have to stop degradation, and the protein is accumulating and enters the nucleus. So key are obviously these enzymes, because they determine oxygen available for the enzymes, degradation, no oxygen, accumulation. So we looked at these enzymes, and we made, these are the proteohydroxylases. There's another enzyme that works similarly, same mechanism, that has only an asparagine as a target. I will, in this presentation, I will keep them together. They both regulate the protein, but we will focus on the proteohydroxylases. So this is what they need: oxygen, 2-oxyglutarate, I told you, and the HIF protein. So we made fusion proteins. We labeled them. This is one of the isoenzymes, 2, 3, and this is the other enzyme here for the asparagine. And as you see, they have different cellular distributions. So the isoform 1 is in the nucleus. The number 2 is actually the strongest in the cytoplasm, and this is in the nucleus and in the cytoplasm. So we were interested in, does this have any meaning? And in particular, is this stable, or is this also something that is volatile? Can the enzymes take different locations in the cell? So we used then an antibody in high resolution microscopy. This is for PHD2, because this is the most active enzyme, and this is a 2-photo microscopy. So you see that the PHD2 is clearly in the nucleus, what we already saw. And now if you look for the Western blot, and this is cell extract, cytoplasm, and nucleus, we found that the PHD2 is under normoxic conditions, is in the cytoplasm, but is also in the nucleus. And it is more in the nucleus under hypoxic conditions. Here is the HIF-1 protein. And what we did in this study is that we also looked for the role of hypoxia. So if you compare this lane here with this lane, you see that hypoxia induces the oxygen sensor itself. That is important for regulation. And we also used nitric oxide in this study. We can go into detail later, because nitric oxide is also inducing this enzyme and therefore interferes with the oxygen sensing process. Now, when we had this information, we determined the activity of the prolyl hydroxylases. And we wanted to know, and this is also work that was done in Finland by Johanna Müller-Harju in an elegant way, and they actually published this before us. So we wanted to know whether the oxygen concentration and the activity of the PHDs fit. And if you look at these activity curves for nucleus and cytoplasm, they are virtually the same. You see that the critical range is exactly the role of, or the range of physiological oxygen concentration in the tissue. So these oxygen sensors work perfectly in the correct range of oxygen concentration in the tissue. And this is actually what we've shown here. This is from a historical study with Helmut Acker. This is the normal distribution of oxygen in the tissue. And so this is the perfect oxygen sensor for regulating genetic responses. Now, some very molecular data I would like to show you, because we are now in the nucleus, and we thought, well, this process, how these proteins find, the alpha and the beta subunits, find each other and then bind to the complex is something we want to understand, we want to study. So this is now in the nuclear structure, and we wanted to do this in living cells. And we did the following. We used our proteins, HIF-1 alpha, and fused them, marked with them with a fluorescent protein. This was either blue fluorescent protein or yellow, so cyanofluorescent protein or yellow fluorescent protein. We also did this for HIF-2 alpha and also for ARNT. And we then used these fusion proteins to use a method called FRET. Probably many of you know this, fluorescent resonance energy transfer, where we have the two labeled proteins. So this would be HIF-1 alpha with its cyano and HIF-1 beta with the yellow. If these proteins are not a dimer, if they are further apart than 10 nanometers, and you excite with a laser the blue fluorescent, then you get blue fluorescent. But if the proteins form the dimer as they do in the nucleus, then the energy from the blue fluorescent is transferred, and then you get yellow fluorescence. So you can discriminate between blue fluorescence and yellow fluorescence, whether you have not a dimer or whether you have a dimer. And if you do this over the whole range, you can use the efficiency by which the yellow light is emitted. This would be totally one protein, 100% efficiency, it's a theoretical value. And then the further they are apart, less energy transfer. Yellow signal becomes weaker. And you can use this as a molecular ruler. So you can determine the distance between two proteins in living cells. And that is what we did. This is a bit complicated slide, but essentially I can guide you through this. This is the nucleus. This is the HIF-1 alpha. This is the blue fluorescence from the HIF-1 alpha cyanofluorescent protein. This is the fluorescence from the HIF-1 beta, ARNT is the other name, yellow fluorescent protein. And this is the combined FRET signal. So in here is the FRET efficiency. The higher the FRET efficiency, the closer the two proteins. And what you can see is that we actually can, in living cells, show, this is the upper curve here for HIF-1 alpha and ARNT, that the cells are 6 nanometer in distance. The absolute value 6 is not so important, but it's less than 10, so they form a dimer. If we do this for HIF-2 alpha, we see that HIF-2 alpha is also forming a dimer, but the distance is a bit more. So I said there are two isoforms, HIF-1 alpha and HIF-2 alpha, and one difference, as we know, is that they form different complexes. They bind to the same DNA element, but they form different complexes. Now, one very important thing from this slide is we also made a mutation. We made a mutation where we cut off, we deleted the part that binds to DNA. So this here is a signal in the nucleus from HIF-1 alpha that cannot bind to DNA. And this ARNT can also not bind to DNA, HIF-2 alpha or not, and this ARNT also not. But as you see, they still form a dimer. So they still make contact. And there's a little difference, and here there's no difference, but they find each other. So it is not important that they bind DNA. They just have to find each other, and through the remaining PRS domain that I was mentioning, they make the dimer. A control experiment is here. This is transcriptional control, normoxic hypoxia, and if they have no DNA binding, they're inactive. So this is the proof that this is really a true dimer. Now, and so this part here I now showed you is not necessarily DNA bound. So this could happen before they bind to DNA. So how can you determine this? Now there is, again, we used the fluorescent fused proteins. And this is a method that is called fluorescence recovery after bleaching. It's also a very elegant optical method. This is one nucleus and the signal you see is HIF-1 alpha fluorescent protein. If you look at high resolution, these are the structures in the nucleus where you see the fluorescence. So somehow these HIF proteins are bound to some structures in the nucleus. You then take a laser and you bleach, so high energy laser, and you destroy the fluorescent proteins in this square. It's under a microscope. So this is a square that is bleached. And then you wait 27 seconds, 51 seconds, and then you see that the structures are coming back because the HIF-1 alpha protein diffuses in the nucleus, it moves in the nucleus, and new proteins, which can give fluorescence signal, go to the same structures they were before. So it's a well-regulated process. Below here is HIF-1 beta. It's the same for HIF-1 beta. So the protein is recovering, the fluorescence is recovering because the protein is moving in the nucleus. And that you can use for this evaluation. You don't have to focus on all the different conditions. Important is all the black solid here are constructs with HIF-1 alpha. There are different mutations and shorter forms. That is all the same. You see there's no difference. But you see the fluorescent that comes back up to 100% over time in seconds. So quite fast, the protein moves in the nucleus and recovers the fluorescent. This is HIF-1 alpha. Now HIF-1 beta. This is all the gray lines. And these gray lines are clearly different from the black ones. And that is good evidence that the two proteins, HIF-1 alpha and HIF-1 beta, are moving separately. Because if they, I go back one slide, if they had the same, if they were one complex, they have to move with the same speed. But because they move with different speeds, they are not in one complex. So the two proteins here are separated in the nucleus, and they find at some place before binding to DNA. So this is still unpublished. We are still working on this mechanism, but this is, we believe, quite interesting with respect to regulation within the nucleus. Now, I'm coming back. I'm coming to my two last slides, and making somehow the connection to medicine and applicability of these basic research findings. If you may remember, I told you that there are prolyl hydroxylase inhibitors, like the substance from Fibrogen, that's the one that is now on the market. And if you inhibit this enzyme, then you inhibit the degradation, and then the protein goes into the nucleus and can initiate gene transcription. And I had already shown you the slide. So China and also Japan have approved these substances to induce erythropoietin production, and particularly in people where the kidneys are sick and they have chronic kidney disease. And I want to show you two slides from the very remarkable initial work also from Bill Kalin's lab, where he showed that this works in a mouse. And what he used in this mouse was luciferase, and that was controlled by the oxygen degradation domain of the erythropoietin gene. So this part that is responsible for HIF degradation, that he used and coupled to luciferase. And this is the mouse from the back, and here are the kidneys, and you see that the kidneys are the most sensitive where the luciferase light is already emitted under normal oxygen. And then if you put the mice in hypoxia, you see that the kidneys here light up because the animal is hypoxic and the ODD drives luciferase. And in those days, they then had the first substances from the first prolyl hydroxylases inhibitors I showed you. And so this is the control, the vehicle. This is the liver from the mouse. And with this dose, you see a clear signal here. But more interesting here is the kidney. So this was very initial work in 2006, almost 11 years after isolation of HIF, that they had already the idea how to use this in therapeutics to stimulate in the kidney, which is the most sensitive organ for this response, to stimulate expression, in this case, the light emitting luciferase by using the HIF system. And this is the, they also showed that the erythropoietin goes up from 290 to 48,000. So this was the proof that the kidneys are still intact and that they can produce erythropoietin. So, and with this more or less application of this basic science of oxygen sensing and the result of understanding how cells sense oxygen, I'd like to end and just acknowledge 3 of the many people. So André Bernardini did all the imaging, and with Jun Hu, who was a postdoc, a doctoral student in my lab with Christoph Watzlaw. So they are the magicians with the microscope and with all these optical methods. And after the Nobel Prize was awarded, we had this group of, I would say, 2nd generation postdocs who more or less spent time in the labs of these people, and we collected these historical stories about this and put this in a review article if you are interested. And with that, I thank you, and I'm happy for any questions. Thank you.
Joachim, thank you so much for your so valuable presentation. I think that oxygen sensing mechanism is one of the fantastic and interesting area because of touching many different topics as you indicated. And before taking a question from my colleagues, I would like to ask one question to you for way of our research program all over the world. Now you talk about interaction, protein-protein interaction and gene-protein interaction. Yes? What is your opinion for future? And you talk about some pro-drugs and enzyme inhibitors. Yes? Mm-hmm. Which topics is important? Protein metabolism or interaction, or DNA metabolism. When you look at your presentation, especially your study, I understand that because you know I'm focusing on protein metabolism, as you know, is enzyme and enzyme kinetics. For this reason, I always focusing on this area. In the future, which one is going to forward? Is protein interaction or gene protein interaction process? Mm. Well, this is your personal question. Yes, okay.
Yeah. I think actually it is both, because as you see from the VHL degradation, so the degradation process, that the main regulation of HIF is the inhibition of degradation. That clearly shows that protein metabolism and the handling of protein is very important.
Yeah.
And there is now, beside hypoxic regulation, there may be other ways how HIF is regulated, and particularly it regulates then metabolism in the cell. The protein-protein interaction is important because there are now the first inhibitors of HIF on the market. Mm-hmm. And for HIF-2, they're in clinic, not on the market, they're in clinical trials. And the mechanism is that they specifically bind to HIF-2. There's a small binding pocket, and they inhibit the protein-protein interaction between HIF-2 alpha and HIF-1 beta. And that is specific. People say it's specific. We are also studying this. This would be specific, and if you can interfere with this protein-protein interaction, you can specifically inhibit HIF. So HIF is so important, and HIF is in all cells, so you cannot just totally inhibit HIF. That we will not survive. I mean, the mouse with the HIF knockout is not viable. So it's so important for every cell. So you have to do this specifically. So the answer is both. The one is the degradation of the protein, very important for regulation and also for different forms of von Hippel-Lindau disease. And currently these these inhibitors, they rely on protein-protein interaction.
Thank you so much. Değerli arkadaşlar, sorularınız var mı? Sorularınızı doğrudan mesaj şeklinde yazabilirsiniz. Profesör Fandrey onları görebiliyor. Ben de görüyorum. Sorularınız var ise onları cevaplayacak. Cevaplamaktan mutluluk duyacak. Onu konuştuk biz oturum öncesi. Evet, sorularınızı bekliyoruz. Katılımlarınız için ben ayrıca teşekkür ediyorum. Güzel bir toplantı oldu. Katılım da iyiydi. Evet, soruları alabiliriz. Joachim, I want to explain something to my colleagues about question. Okay.
I have one question in the chat, which is interesting.
I couldn't see it, but okay, you can.
So the question is,
you can explain. Okay.
Yeah, the difference between HIF-1 alpha and HIF-1 beta. So as I said, HIF-1 alpha and also HIF-2 alpha, they are regulated by oxygen, and HIF-1 beta is not regulated by oxygen. And I know that there is one or two papers, they think there is some regulation. This is all compared to the manifold regulation of HIF-1 alpha and 2 alpha. This is negligible. So the regulation of the HIF complex is through HIF-1 alpha, definitely. The other question is why prolyl hydroxylases is in the cytoplasm, and I've gone over this a bit fast because this is a complicated story. The cartoons in the reviews, they always paint the prolyl hydroxylase in the cytoplasm. However, I showed you data that prolyl hydroxylase is also in the nucleus, and we have measured the activity in the nucleus. Now, this is physiologically, this is a bit interesting, a bit complicated, because you know that the mitochondria around the nucleus, the ER is around the nucleus. So if the oxygen sensor is in the nucleus, you somehow have to claim that the oxygen has to go through all the structures in the cell and is then ultimately registered in the cell nucleus, which is maybe, but this is still controversial. So I really can't say. When we did cell fractionation and immunohistochemistry with specific antibodies, we see that prolyl hydroxylase, the hydroxylases are also in the nucleus. Interestingly, von Hippel-Lindau protein initially was described in the cytoplasm, and that was because people thought then that HIF protein goes out into the cytoplasm again, and then is degraded by von Hippel-Lindau in the cytoplasm. But now there's also indication that von Hippel-Lindau is available in the nucleus. So that is, well, and then the last question is about erythropoiesis and HIF-alpha. I don't know whether this question is touching this controversial issue, whether erythropoietin is regulated by HIF-1 alpha or by HIF-2 alpha. In the mouse, and this is now the current knowledge that you probably have to, you should quote this when you write a paper. In the mouse, EPO is controlled by HIF-2 alpha. However, when we looked into human cells, primary human liver cells, we also see that erythropoietin is regulated by HIF-1 alpha. We are still working on this. There may be a species difference, but I would now say the consensus is HIF-2 alpha regulates erythropoietin, and HIF-alpha regulates, for example, glycolytic enzymes. So adaptation under hypoxia, glucose, anaerobic glycolysis, this is regulated by HIF-1 alpha.
Thank you. Thank you, Joachim. Dear friends, if you have more questions, you can write directly to Professor Fandrey. Yes, Joachim. And again, I want to thank to all of you. And Joachim, I
have two more.
I hope.
If I may, I have two more short questions where they ask for 1 or 2 sentences. If I may, 1 or 2 sentences.
Yeah, we have one question. Yes, you say true.
Okay, so
yes.
The advice for young researchers is really be fascinated and do what you find interesting and work you like. That is absolutely important. That is what I also tell my PhD students. And you may have luck and you end up in a field of Nobel Prize, or you may end up in a very interesting field of immunology, for example. I believe immunology, not only because of COVID, but also because of tumor immunology, is a very interesting and hot topic. And for the reactive oxygen species, if you may, you can look up one of our papers. Reactive oxygen species may be modulators. They are not the key sensor molecules. I have had many fights with the ROS people, and I started my career with ROS, and the protein hydroxylations I told you are much faster. The ROS is much slower than the HIF hydroxylases.
Yes, that's true. I remember, Joachim, our study about free radical metabolism and enzymatic processes. You say true. Yes, thank you. I hope that we will meet in Duisburg or Ankara face-to-face after COVID infection. Thank you.
Yes, thank you.
Joachim, again. Hope to see you.
Okay, yes, thank you.
Bye-bye.
Bye-bye.
Bye-bye, thank you so much.
Hocam kapatıyorum oturumu.
Kapatalım, evet.
İyi akşamlar.
Ben teşekkür ediyorum. Bir dakika, 33 kişi var toplantıda. Dışarı Profesör Fandrey ayrıldı. Tabii kendisi özellikle alanda bahsettiği gibi önce serbest radikal metabolizması, daha doğrusu ikimizin de hocası olan emekli oldu. Wolfgang. O da alanda çok büyük bir hocaydı. Kendisi daha sonra üniversitesini değiştirip bahsettiği gibi SNDS geçti. Alanda bir miktar değiştirdi. Gördüğünüz gibi çalışmaların büyük bir çoğunluğu moleküler seviyede yürüyen çalışmalar. Tabii içinizden özellikle ben bu konuyu biyokimya ve fizyoloji derneklerine de duyurdum. İçinizde belki konuya ilgisi ve alakası olanlar vardır. Kendisinin e-maili sayfasında mevcut. Doğrudan yazışabilirsiniz. Veya soracağınız sorular olursa cevap verecektir. Kendisinin tabii çok fazla idari rolü var. Söylediğim gibi hem enstitü müdürlüğünü yürütüyor hem dekan yardımcılığını yürütüyor. Ama alanında önde olan araştırmacılardan bir tanesi. Ve Nobel ödülüyle ilintili de tabii çok yakın çalışıyor özellikle HIF'la ilgili. Ben tüm katılımcılara, tüm meslektaşlarıma ayırdıkları zaman için teşekkür ediyorum. Bu toplantılara aylık devam edeceğiz. Amacımız yine alanında böyle tanınmış bilim insanlarını sizlerle yan yana getirebilmek. Çok kolay olmuyor ama yine de buna çaba göstereceğiz. Ben iyi akşamlar diliyorum. Görüşmek dileğiyle. Teşekkür ederim size.
Rica ederim hocam. Katılımcılarımız da çok teşekkürlerini iletiyorlar. Mesajlar bana geldiği için siz göremiyorsunuz.
Ben hiçbir şey göremiyorum, evet. Tamam. Olur.
İyi akşamlar herkese.
İyi akşamlar. Sağ olun. Sağ olun.